Hi everybody, my name is Katherine and I'm from Jonathan. Last year we went to the University of New Hampshire and we worked with Dr. Suresh Manocha on plant cloning, and part of our project was to understand asexual and asexual reproduction of cloning of plants. For our objectives, our professor wanted us to plant clones, and what we did was he set up an experiment for us, and we just followed the procedures to how we clone the plant.
So just some background information. John everybody took biology. There's asexual and sexual reproduction. And next slide. The materials we use are listed over there if you want a copy of them, but it's not necessary.
For the sexual reproduction, they're done naturally by plants, and that's when there are also auxins and cytokinins. Auxins are plant hormones, and it causes the lengthening of cells. Cytokinins are plant growth substance. And meiosis is a cell division. Asexual reproduction is also known as cloning, which is what we did.
So,
I think it's a good idea to explain that, go back to the previous slide and explain that with a pointer.
Asexual reproduction is cloning and what we did for this was that we took the plant and we just cut it up and put it into the what our professor had prepared for us, which was a median. It was inside a little tub that when the plant touched it, it would just through the chemicals it would start reproducing itself.
Alright, so you saw before, clone is basically an exact genetic copy of an organism. So for our plants, we took a cell and we just replicated it, or induced replication. Alright, so this is actually much easier than cloning animal cells, since you can do it from just a single cell. So all you have to do first is just remove a plant sample. What we had was, we had a plant and we had a scalpel and forceps, like in the picture, so we just removed the sample from the leaves. And we placed that sample onto an agar plate, which has nutrients necessary for growth and hormones of desired. Auxins would stimulate roots to grow, and cytokinins would stimulate shoots. And so that's an example, but that's more of what it was is we kept the medium in motion, so that's more of like an amalgam of just undifferentiated cells.
Use a pointer. What are these things?
Those are the cells over there. So it doesn't look like it, but it's because how Dr. Manocha put it, he said we confused them by using a mixture of hormones.
You have to describe what it is. This is a beaker, right?
Yes.
That's actually a glass bottle that the plant samples were put in, and what happened was that
What's the bottom?
the bottom is actually the medium that was used to help the plant cloning process.
What is the medium?
He actually didn't go through that because it was already pre-made for us. Because
It's important to know what the medium is.
yeah, but the thing is we didn't have enough time, so he just.
Well, you have to know.
So again, these are just pictures of different samples he showed us. That's the agar plate. Those are the plants. You can see there are a lot of shoots here. Not so much shoots. So yeah, after you've done that, you place it on the agar plate. All you need to do is just give it some sunlight. And so That's the room. See, this was actually in constant motion. That's why there was that mushy, just massive plant cells just off the floor. Over there are the just normal plates.
Okay, so like I said earlier in the lab, we cloned out the plants, and they were stored inside a controlled environment for about 5 days since we were there. And the results, depending on how we did it or maybe how the medium was created, was different for every one of us. So there were 2 possible results. One result would be that the clones would be, the plant cloning would be successive, which means that plants would actually, like more leaves and stems would actually grow. In other cases, like if it was a failed cloning, then you will see beans go in tumors from plants. I think most of my plants that I cloned had gained tumors, which looks like the picture from before. So in the future, we hope to continue our project in plant cloning and hopefully work with different plants instead of just normal plants that we would find in the park or something, but more like flowers or something else. These are references in our account. What went wrong was probably the medium or maybe how you cut out the plant, because there are different, the way how each plant was cut out depending on how the cells were, then